Okay, so I just gave you a start on cellular respiration. So last lecture, just to remind you, of course, that week from tomorrow will be the first exam, and that exam will be in lab. So we'll have a Monday lab. Next week there'll be no lecture quiz. Okay, because the policy is that on those weeks that we have a lecture exam, then there's no lecture quiz. So there will still be lab quiz on Monday, okay? And on Wednesday, there'll be no lab, just only the exam, okay? That's all, okay? We'll use it in the lab period. It'll cover lectures 1 through 5. Cellular respiration, photosynthesis will not be on it, okay? Just on the metabolism. And so I uploaded last year's exam. It's up there, and the best way to use that is to study everything first, make sure you know it, and then sit down as if it is a regular exam. Take it like a regular exam. Limit yourself to maybe 2 hours and 20 minutes, because usually that's what we give people, 2 hours and 20 minutes, thereabout. And don't look at your notes, okay? Take it as it is. And then over the weekend, probably Friday, late Friday, I will upload the answer key. Okay, now my answer keys aren't just only the right answers for some of the harder questions. No, for some of the questions that's all, because you should know that. Okay, it's very obvious. And so sometimes multiple choice, I just say the answer was A. Okay, but in other cases, I make explanations as to why. Okay, so I reason through it and write. So it's not just only an answer key; it's also explanation. All right, how it should go. All right, then check yourself. Okay, and then see what you know, see what you don't know, and for studying and preparing for the exam, concentrate on the stuff you don't know. Okay, maybe review the stuff you know, but concentrate on what you don't know. Okay, that's the best way to use it. Also, another thing with MindTap. Okay, notice that there's some people not doing the MindTap exercises. And you're really hurting yourself, because those are the easiest points you'll ever get in this class. Okay, now, even though it's past some of these, you've left them 3 weeks ago they were due, I still will forgive and extend 2 chapters. Okay, 2 chapters only. Okay, and so if you get your act together and you get into it, just send me an email for the 2 chapters you want extended, and I'll give you an extension on them. Once I get your email, once I respond to your email, you have 24 hours to make it up. Okay, just to let you know that no matter how far back, I will extend to. Okay, getting into cellular respiration. Okay, so I gave you a breakdown here. This is the summary equation. Earlier, when I was going over metabolism, I did introduce this, and I said that you didn't have to know it at the time, but now that we've hit this particular screen, you do, okay? You have to know this formula. This is for a pathway, okay? Some parts of this pathway does have reactions that most of them are exergonic, but there's a few endergonic reactions too, but the overall outcome of this pathway is that it's catabolic. And therefore, at the end, under standard conditions, the free energy change is minus 686 kilocalories per mole. That was a number I said I want you to know, because I won't give it to you on quizzes or exams. It will be something you have to know. All right, now, when you're doing these standard free energy changes, it's very important that you put a plus or a minus in front of the number, okay? Because if you don't put a plus or minus in front of the number, just the number, I'm going to assume that it is a positive number. Okay, but there's a big difference between whether it's exergonic or endergonic, and it has to be stated that way. Okay, so this is the summary equation, and the whole function of cellular respiration, as we shall see, is to regenerate ATP. ATP from ADP plus phosphate, inorganic phosphate. And this is another number that I wanted you to know. The free energy change for both the hydrolysis of ATP and for the regeneration of ATP. Okay, under standard conditions, it's the same magnitude, 7.38 kilocalories per mole, but if it's the hydrolysis of ATP to ADP plus phosphate, then that is a minus sign, okay, because it is exergonic. For the regeneration, which is the whole point of cellular respiration, it is endergonic, okay? And where the energy comes from ultimately is chemical energy that's stored in the food, okay? Chemical energy that is stored in the food, slowly being degraded, captured, and used to generate ATP, all right? So I went over oxidation reduction, so I'm not going to go over that too much more here. Just that this is the movement of electrons in the form of hydrogen atoms, okay? And so in biological systems, to be able to tell oxidation versus reduction is quite easy because always the reduction involves removal of hydrogen, and always the, oxidation, the removal of hydrogen, reduction is the binding of hydrogen. So that's what's going on here. So in cellular respiration, it's the glucose that is being oxidized, and it is oxidized to carbon dioxide and water. What is being reduced is the oxygen that we breathe, and it is ending up as water. Water has more free energy content than the oxygen that it started out with, okay? The more oxidized it is, a compound is, or substance is, the more oxidized it is, the less free energy it contains, okay? All right, so this is due to the transfer of hydrogen ions, all right? Okay, and let me see here. I believe we're pretty close. This is where I probably stopped. And so I'll continue. I didn't even make it over to the slide. All right, so these electrons and hydrogens are not simply transferred directly from the food substrate to oxygen. Okay, there's an intermediate carrier. So this is called nicotinamide adenine dinucleotide, and it is sort of a shuttle for those electrons. All right, you just only need to know it as NAD. This comes in two different forms: oxidized form, which is NAD+, and a reduced form, which is NADH. Okay, all right. So this is a nucleotide derivative, as I'll show you here in a moment. So it's an energy carrier. It is a coenzyme, all right? So it's a coenzyme that's derived from the B vitamin niacin. This is one of the reasons, one of the main reasons you have to have niacin in your diet. We cannot make NAD without having that precursor molecule, so we have to get it in our diet. And this is a coenzyme that's found in a collection of enzymes that are called dehydrogenases, okay? So this is a class of enzymes, dehydrogenases, and what they do is they catalyze hydrogen transfers. Hydrogen transfers. That's why they're called dehydrogenases. And so, although this is the one and the same, nicotinamide adenine dinucleotide as a coenzyme, it is still at the active site. It can move into the active site and out of the active site of these dehydrogenases. So it's fluid. But nonetheless, it has to be bound to the active site of a dehydrogenase in order to do this. And so it assists in stripping the hydrogen from the substrate. And so this is an oxidation reduction reaction, and therefore NAD+, the food substrate becomes oxidized. And NAD+ is reduced to NADH plus a hydrogen ion, okay? So in here, we're talking about two hydrogen atoms, okay? Two hydrogen atoms. Hydrogen atom consists of proton and electron, right? Proton and electron. So we're looking at 2 protons or hydrogen ions, 2 electrons. All right, so NAD can take 2 electrons and 1 proton. That leaves another proton into the surroundings, okay? And I'll show a picture here of what this looks like. All right, so it is a dinucleotide. That means that there's 2 nucleotides. One of them is adenine with ribose and a phosphate. That's adenine riboside. And so we've already familiar with that. Number 1 carbon, number 2, 3, 4, 5, and here's 5 carbon. We have a different nucleotide that we haven't come across before. Indeed, it has ribose, as typical, but at the number 1 carbon, it has a different type of nitrogen base called nicotinamide. Okay, now this nicotinamide is a nitrogen base, but it is not the type of nitrogen base we find in DNA or RNA. So there's 2 nucleotides there, okay? And so this is the oxidized form. And the reduced form, what happens here, notice we have a double bond here on the nicotinamide. That can take another whole hydrogen, okay? Proton plus electron. And there's this nitrogen down here. You notice that nitrogen that's connected to the number 1 carbon on the ribose, that has a positive charge. It can take an electron and become neutral. And that kicks off, it's not shown here, but leaves over a hydrogen ion, which is given off into the surroundings, all right? So this is what NAD plus happens to work with. So summary formula down here, 2 electrons, 2 hydrogen ions, which constitutes 2 atoms of hydrogen, okay? 2 atoms of hydrogen. Okay, so this is sort of an adapter. So what's happening here, we have a food substrate, 2 electrons or 2 hydrogen atoms are stripped off of it, transferred partially to NAD+, so that we now have something that is reduced, or excuse me, oxidized. The food substrate is oxidized, and we have reduced NADH. Okay? All right, now, okay, so that represents a loss, and NAD is the reduction, it's the gain of electrons in the form of hydrogen, okay? Now, probably jumping ahead here, but I'm going to go ahead and do this. Okay, the more hydrogen you have, the more the more reduced it is. Therefore, we have here, if I just do an aldehyde, or maybe a fatty acid, all right? I won't worry about this. Okay, so for a sugar that has the most, we now oxidize that. Let's go ahead and oxidize this right here. Now we have ketose. So first of all here, it was a, no, let's go one more. Let me go one more. Well, let's start over. If we have, I'm looking at this 1 carbon right here. Okay, I'm just going to, if we have carbon, has all those hydrogens on it. Okay, that carbon is more reduced, then has more free energy than that. Actually, let's do one step more. Keep that there. It's now, it went from sort of just only having hydrogen, that's all, to having an oxygen, but we still have 2 hydrogens, all right? So, but this here, because of the hydroxyl group, that is still more oxidized than the first one, It contains less free energy. Then if we could take it one step further, then put up our 3 here. Actually, if we do that, we'd have to split it. No, do that. Put it into a ketose, and that is even more oxidized, less free energy. There's a series like this. And if I took it one step further to make this carbon a carboxyl group, then it's even more. We'd have to split that off, all right? So we go down to here. And we're making that carbon into a carboxyl group, and therefore we had to split off that end. It has to be transferred somewhere else. So my whole point, carbon with 2 hydrogen, highly reduced, and this with the hydroxyl group has less free energy, it's more oxidized than the first. This here with a ketone function, carbonyl function, it is even less free energy, and a carboxyl group is most oxidized. What does this sort of look like? It looks like carbon dioxide, inorganic carbon. If we cleave it off, we have carbon dioxide, which is even more oxidized. All right, so that's a reduction energy series, and you're going to see it as we go through some of this cellular respiration. Okay. All right, so moving on. Cellular respiration has 4 phases. Something called glycolysis, in which it picks up some types of, can produce some ATP, some pyruvate oxidation. Krebs cycle, this is also called the tricarboxylic acid cycle. It's also called the citric acid cycle. It's called a lot of things. I think your book calls it the Krebs cycle, and that's what I'll do, yes. The further you take the hydrogens away from that carbon, the less free energy it has. Because of its potential energy, it's because of the location of those electrons with regard to the carbon nucleus. When you put that oxygen in there, it's pulling the electrons away from the carbon. Okay, so it's not available to the carbon anymore. It's a loss of electrons. Okay, all right. So Krebs cycle produces something called electron transport chain. So I'm going to go through all these phases and show exactly what happens. Glycolysis is actually 10 steps, okay? 10 enzymatically cataloged steps. Pyruvate oxidation is 3 steps. Krebs is quite a few steps, and then also electron transport chain is quite a few steps. And so going through each one of these, notice here that there's no ATP produced in pyruvate oxidation, but in the Krebs, most of the ATP is produced here, electron transport chain, which happens in the mitochondria, and I'll get to that momentarily. But I'm going to start with glycolysis first. This is the only part of cellular respiration that happens in the cytosol. Okay, and it is an ancient pathway. We know that goes back billions of years, okay? And it's pretty much universal in all living things. The enzymes that are involved here, would they have been produced on a free ribosome or a bound ribosome? Free ribosome, because these are happening in the cytosol, okay? This is not in any of the endomembrane systems, so it's in the cytosol. This is anaerobic. It doesn't require oxygen, okay? So oxygen is used somewhere else. Oxygen is necessary for cellular respiration, but not for glycolysis. Glycolysis can do that. Now what happens here, I want you to keep track of the carbons, okay? So I'm just putting up here C6. We're going to start with glucose, because glucose is the preferred energy source. Okay, other things can be used, but I'll come back to that later. So during glycolysis, we take glucose, and you know we have several carbons there, 6 carbons, and there, through partial oxidation, we end up with 2 3-carbon entities called pyruvate. Okay, pyruvate is this food substrate at the end of this glycolysis. We still have all 6 carbons. What happens here is that we will extract some of the energy from the glucose, and midway through we split that 6 carbon entity into two 3 carbon entities called pyruvate. Okay, and there's 2 different phases to this. There's an energy investment phase and an energy yielding phase. Okay, the energy investment phase happens first, and the energy yielding phase comes next. So some of the steps here, okay, starting up here, we have glucose, and there's an enzyme hexokinase, and glucose 6-phosphate, and all these are the enzymes that are involved. These are the structures. Once we change something on glucose, it's no longer glucose, it's something else, okay? Keep going through here. So by the time we get to the next exam, I expect you to be able to know each of these steps, draw each of the compounds, know all of the enzymes involved, and this is just the beginning, okay? We still have everything else to go through. Yeah, some of you are laughing. Yeah, you know that I'm just bullshitter. All right, so, okay, yeah, no, you don't have to know all that. Okay, so what do I want you to know? Now, I'm going to go into far more detail than what I want you to know. For each of these phases, I want you to know the beginning substrates and the ending substrates. Okay, the names, you don't have to draw them. I want you to keep track of the carbons. Okay, I want you to keep track of what we get out of it, what we have to put into it, any energy costs and energy gain. So you're going to keep track of the energy carriers such as ATP, nicotinamide adenine dinucleotide, and one other that I'll introduce a little bit later. So that's all I want you to know. The cost, what you get out of it in terms of energy, beginning substrate, ending substrate. Okay? And there's just a few tiny details in between them I can have you do, but that's all. Okay, so I'm going to have my simplified version. If you want to memorize this, that's great. Okay? If you go on to the university and take a biochemistry class, yeah, you will. Not only would you have to know phosphofructokinase, but a lot of times you have to know the mechanism by which the enzyme happens to work. Okay, so, but we're not going to do that here. Okay, so glucose. First of all, we have to phosphorylate it into 6 phosphoglucose. Okay, so this requires 1 ATP, so we're attaching a phosphate to the glucose, and now it is glucose 6-phosphate, okay? And then we have to do it again. So that now it's called fructose 1,6-bisphosphate. You don't have to know that, but I'm putting the name up there, all right? So what's going on here? Isn't cellular respiration supposed to regenerate ATP? And here we're using ATP. Why? Think about your quiz yesterday, or not yesterday, the day before. No, it was yesterday, today's Tuesday. I am totally lost in time. Yeah.
You're destabilizing the molecule, so it's easier to bring apart nonsense.
Just another answer I'm looking for.
I was gonna say energy coupling in a way, because
Say what?
energy coupling, so you're using some.
No energy coupling here.
You are getting some negative free energy into the system so that it can be used, like you can counteract it with some positives later. My brain cannot explain that right, but it was in there.
What is that? There you go. Okay. So, okay, yeah, and so that's part. Okay, there is a little bit with destabilization too. All right, so then at the end, this breaks in half. Okay, there's a couple of other steps here, rearrangement. Breaks in half, and we now have 2 molecules of glyceraldehyde 3-phosphate. Okay, so I want you, even though this is the end of the energy investment phase. And I do want you to know in substrate of the energy investment phase, okay? So there's two parts here. We begin with glucose, 6 carbons, and we end with 2 molecules of glyceraldehyde 3-phosphate, and it costs us 2 ATP, okay? You need to keep track of the cost as well. So in each of these phases, the ending substrate is the beginning substrate of the next phase, okay? So the beginning substrate of the next phase here, namely the energy yielding phase, is 2 molecules of glyceraldehyde 3-phosphate. So keep track of how many we have, all right? Because we have 6 carbons still. The 6 carbons originated from the glucose, okay? So energy yielding phase, glyceraldehyde 3-phosphate, and we get our first reduction of NAD+. Okay, I have 2 of them up there because we have 2 glyceraldehydes 3-phosphates. And so hydrogen atoms have been passed over to reduce to NADH. And furthermore, coupled with this is a step where inorganic phosphate is now put in. We have 2 of them, 1 for each of our substrates. We have 2 of them, okay? All right, keep track of that. So we're attaching a phosphate to our substrate. So once again, we have a 3-carbon substrate that has 2 phosphates on it. Well, putting those phosphates on there is inorganic. So how does it happen? It's inorganic. How can we put phosphates attached then covalently to the substrate? Where did the energy come from? We had to put it in there.
The reduction reaction?
Say what?
The reduction reaction for the,
Exactly. Example of energy coupling. It's not always ATP, okay? This here is exergonic. And that pays for the inorganic attachment of that phosphate. So once again, we have an inorganic and an exergonic coupled reaction coupled together. Okay, and so this is what provides free energy for phosphorylating that substrate. Okay, and what catalyzes this step is called triose phosphate dehydrogenase. Now, you don't need to know the name of this enzyme. My point is, it is a dehydrogenase. Okay, I do want you to know that these steps where we are reducing NAD to NADH involves a dehydrogenase, and the NAD plus is a coenzyme at the active site of that dehydrogenase. Okay, so this is a group of enzymes that I do want you to know. This particular dehydrogenase only catalyzes this step. There are other dehydrogenases that catalyze other steps, okay? But they're all common in that they're involved in hydrogen transfers from our food substrate to NAD plus to reduce it to NADH, okay? And then we pass one of these phosphates onto ADP. Because now what we've done here is, for one thing, we have elevated at the very beginning in the energy investment phase by putting those phosphates onto ATP, overcoming activation in a barrier. We have also elevated the energy content of that glucose. Okay, so it's a higher free energy, and so now we can we have enough energy stored here that we can pass a phosphate, one phosphate. We have 2 of them, 2 substrates onto 2 ADPs to produce our first 2 ATPs. Okay, and then we do it again. Okay, happens twice. All right, and this is at the end. We now have 2 molecules of something called pyruvate. Right. All right. So notice this carboxyl group. Okay, as I said, that looks pretty much like if we cleave it off, we have carbon dioxide. All right. Okay, so a summary here. Now, the pyruvate is at a lower energy level than our free energy level than our glucose. However, it's not completely oxidized. We still have some energy content there. So to summarize this, okay, this is what I want you to know, is a cost to ATP per glucose. And what we've gained is 4 ATP and 2 NADHs, okay? That's where we've gained some. So our net outcome is 2 ATP, because we have to pay back, okay? We got 4, but we had to use 2, and therefore our net gain is only 2 ATP, all right? That's all. Not much, all right? And now, as I go through here, I'm going to mention that ATP is generated by 3 different mechanisms. 3 different mechanisms. This is the first mechanism. Okay, it's called substrate level phosphorylation. Okay, substrate level phosphorylation, because where did that phosphate come from that was used to produce ATP from ADP? It was attached to the food substrate. Okay, so there's a binding site. So for these enzymes, this is you don't have to worry about that. But this is phosphoenolpyruvate dehydrogenase, or not dehydrogenase, it's something else. But nonetheless, don't worry about it. You don't need to know the name. It has a binding site for ADP, and right next to it is a binding site for the food substrate. This, in this case, is phosphoenolpyruvate, and it has a phosphate. So that phosphate is put in the active site right next to the phosphate that's on ADP, and we have enough energy in here to form a covalent link, okay? So this is substrate level phosphorylation. All right, now all this is happening 10 steps in the cytosol. Now the pyruvates are taken into mitochondria. The rest of the action here happens in the mitochondria, okay? So the pyruvate goes into the mitochondrium, and one of the first things that happens is that carboxyl group is cleaved off as carbon dioxide. We have 2 of those pyruvates, okay, because we have to keep track of all the carbons, all the entities that originated from that original glucose. And so now, don't worry about the name of this, because now our food substrate has how many carbons? Our food substrate, we have two of them. I'm talking about for 1. How many carbons does one food substrate have? It started with 3.
Oh, now it's 2.
2, okay? So we've lost 1 carbon from each, okay? So that's what, and this action is catalyzed by another group of enzymes, okay? But it's very specific, but we have a lot of these that catalyze different steps. This is called a carboxylase. Carboxylases are involved in carboxyl group transfers. And this is not going to be the first carboxylase we come across. Okay, there's others. But this is a carboxylase. And yes, I do want you to know what carboxylases are and what they do. Okay? Carboxyl group transfers. Then we get another oxidation reduction where NAD is plus is reduced to NADH. Why do we have 2? Because we have 2 substrates. So 1 per each substrate. And then there is, this is here, this enzyme is pyruvate dehydrogenase. You don't have to know, I'll never ask you about pyruvate dehydrogenases, but I could ask you that what is the general group of enzymes that catalyze that step in pyruvate oxidation, okay, which is a dehydrogenase, the whole point. Notice here that this is only catalyzes this particular step. There was another dehydrogenase up in glycolysis, catalyzes those steps. Then, another nucleotide derivative, another coenzyme called coenzyme A. It is, you attach that 2 carbon entity that is left over here is now covalently attached to it, and what we have here is acetyl-CoA. This is acetyl group. It should be something you should recognize, because I mentioned that acetyl groups was something, one functional groups that I wanted you to know. So this is acetyl-CoA. We have 2 of them, right? Okay, because how many carbons in total do we still have there? And acetyl-CoA in both of them. 4, okay? We're still dealing with 4 of the carbons from glucose. All right, and so this is the shuttle that goes into, and I'm not gonna worry about the enzymes involved here, but bear in mind, there is an enzyme in each and every step, okay, along the way here. And so these are then shuttled into the Krebs cycle. The Krebs cycle is a cyclic pathway, okay? And it's not gonna be the only cyclic pathway we come across. There's gonna be others. So what is a cyclic pathway? A cyclic pathway is a pathway in which one of the beginning substrates is regenerated at the end. Okay, we get it back. We don't necessarily get back all of the beginning substrates, but at least one of them we regenerate at the end. Okay? Now I'm going to give more details here, once again, than what I want you to know, because what I want you to know for the Krebs cycle is the beginning substrates, the ending substrates, and all of the energy carriers that we get out of it. Any cost, should there be any cost, and keep track of the carbons. Okay, that's all right? You just need to know the black box, all right? But I'm going to go into more detail. One of the big beginning substrates is a 4-carbon sugar called oxaloacetate. Okay, this is a carbohydrate. All right, oxaloacetate, 4 carbons. We have 2 of them. From the previous step, the pyruvate oxidation, we have 2 acetyl-CoAs. So those 2 acetyl groups are attached. We have 2 attached to the oxaloacetate, one each, and it gives us something that has 6 carbons, okay? All right, so the beginning substrates here, oxaloacetate, acetyl-CoA, that's the beginning. And I would want you to know this result, citrate. This is why this is called the citric acid cycle. Okay, citrate, citric acid, here we're talking about base form, acid form. That's very common, all right? Just like you would have an oxaloacetic acid as well, okay? But this is the ionized form. All right, so what happens here, I'm gonna go in more details, but we'll summarize this again. Then we cleave off 2 more carbon dioxides, because this here entity, citrate, contains a carboxyl group. Therefore, we can cleave it off, all right? So that represents now we've lost 4 of our carbon dioxide from glucose. So now the substrate, how many carbons do we have? 5, okay. We now have a 5-carbon entity, and we have another reduction here. Another NAD, 1 for each of our substrates. Hydrogens are passed on, and now we gain 2 NADHs. Then we cleave off, because that produces another carboxyl group, okay? So this reaction produces another carboxyl group, and then that is cleaved off. Now how many carbons do we have? 4. Okay, now, how many carbon dioxides have we lost up to this point in total? 6. Okay, now those 6 carbons represent the 6 carbons in glucose, but not their identity is different, because really we have to go through 2 cycles to get rid of the actual carbon dioxides that come from glucose. So really some of the carbons that came from glucose are on the substrate. But I only want to do bookkeeping here, okay? We had 6 carbons in glucose, now we've lost 6 carbons, okay? That's all I'm getting at, all right? Not the actual thing. All right, so that means our substrate is 4, all right? And so we get another oxidation of NAD plus to NADH. And then another step, where once again, there's some details I'm leaving out, where we get attached phosphate to our substrate by energy coupling. Okay, I'm not showing some of the reactions, but there was a exergonic reaction. This here, attaching phosphate, is endergonic, okay? And so, and notice that this doesn't involve ATP directly, all right? So there's other ways to couple it. Then we reduce, or actually we phosphorylate not ATP, but guanosine diphosphate. guanosine diphosphate. We came across that with the G protein linked receptors, okay? But this is also an alternate energy carrier like ATP. And its free energy change is very much like that of ATP. And so here we're passing the phosphate from food substrate onto GDP, and now we have two GTPs. All right, but for purposes here, bookkeeping purposes, I want everything in terms of ATP. So what we're going to do is easily pass that phosphate from GTP onto ADP so that our outcome is actually 2 ATP. And this here happens pretty much without any loss of energy, okay? So we're counting this. Still, I want you to know that we do produce GTP at this step, but in our record keeping, because I want you to keep track of, as I said, what we gain, we're going to put it in terms of ATP. All right, and we still have 4 carbons, and this is produced by substrate level phosphorylation. Okay, that ATP because the GTP, because the phosphate originated on our food substrate, it was passed directly on to GDP, produced GTP, substrate level phosphorylation. Then we get another reduction of a different energy carrier, flavin adenine dinucleotide. Okay, it's also a dinucleotide, and it has another base, a nitrogen base that we haven't come across, flavin. Okay, I'm not going to show a picture of it, but nonetheless, this here can take, unlike NAD+, this here takes 2 whole hydrogens, 2 electrons, 2 protons. And therefore, this here is the oxidized form, okay, FAD, FADH2 is the reduced form, okay? And this, then, we still have 4 carbons. We still have energy there in our substrate, and therefore we extract it. Once again, NAD+, and after this step, we're back at oxaloacetate. We're back at oxaloacetate. Now, where did, in our equation, if you go back to the equation for this cellular respiration pathway, there were 6 molecules of water on the left side as a reactant. Because, you know, where are all these hydrogens coming from, okay? We have some reactions here that we're not going to go into details about where the water is being placed onto the substrate, and this is the source of some of the hydrogens and so forth. Okay, so let's summarize this. What do I want you to know? All right, okay, let me go back. Let's go back here. I want you to know the beginning substrates, 2 oxaloacetates and 2 acetyl-CoAs to form 2 citrates. And so I want you to know that part. What's the ending substrate? 2 oxaloacetates. Okay? What do we get out of it? When the starting material is glucose. When the starting material is glucose, we get 2 ATPs by substrate level phosphorylation. 6 NADHs. And 2 FADH2s. Okay? Those are the things I want you to know. And we lost 4 carbon dioxides. Okay? It's all there. All right? Okay, now we have completely oxidized our glucose as far as the representative carbons. Because really, 4 of these carbons here in the end substrate actually came from our original glucose, right? So basically, we've been stripping off carbon dioxide from the oxaloacetate, okay? But still, to keep straight, we're just going to call it these carbons represent what was in glucose. Okay, just bookkeeping, not actuality. All right, so for each molecule of glucose, these are the energy carriers. Okay, ATP, NADH, and flavin adenine dinucleotide. And so in glycolysis, the net, we got 2. We actually made 4 ATP, but it costs us 2 ATP to get started. And therefore, our total gain is just 2 ATP. And we also got 2 NADHs from glycolysis. For pyruvate oxidation, we just came up with 2 NADHs. And then in the Krebs cycle, we gained 2 more ATP by substrate level phosphorylation, 6 NADHs, and 2 flavin adenine dinucleotides. And so the total that we have here, 4 ATP, 10 NADHs, and 2 FADH2s. Okay, that's what we have. So the goal of cellular respiration is to make ATP. We don't have a whole lot of ATP here. Okay, we didn't get too much. However, we made quite a bit of nicotinamide adenine dinucleotide and flavin adenine dinucleotide. This is where most of the energy is. These are strong, these are highly reduced, and therefore they're reducing agents. What it means is that they can easily, very easily, pass their electrons onto some other compound and reduce it. All right? So, and this is where most of the energy is. So the whole point of the electron transport chain, our last stage, is now to extract the energy, stored energy in nicotinamide adenine dinucleotide, flavin adenine dinucleotide to produce more ATP. Okay, and it's a little bit of a complicated process. So this here is called the electron transport chain. What this is a series of electron carriers that are in the inner mitochondrial membrane. Okay, so these are electron carriers. Some of these electron carriers carry both electrons and protons, in other words, full hydrogens, and some only carry electrons. Okay, but they're arranged very systematically in a chain that is in the inner mitochondrial membrane. One of the reasons why the inner mitochondrial membrane is highly folded is because it creates a large surface area. We need a large surface area for a lot of these electron transport chains. So our electrons, which are now stored in NAD, FADH2, are passed into the electron transport chain, passed from electron carrier to electron carrier to electron carrier, down an energy gradient, because each time those electrons are being passed, they're losing free energy. Okay? They're losing free energy due to their location with regard to the nucleus. Okay, so something that's held further away from the nucleus might, in this case, closer, has more energy, and as it's pushed out, it loses energy. So the final electron acceptor is the oxygen. This is why you breathe. Okay, if you don't have that, then the whole electron transport chain gets saturated, and you don't produce anything. And I'll come back to that here in a moment. Okay, and so how does that produce ATP? It's a little complicated, so I'm going to go through this a little bit slowly. Because the passage of electrons down the electron transport chain is incrementally releasing small amounts of energy, which can be used to do work. And that work is to create an electrochemical gradient across that membrane. That electrochemical gradient is an energy source. And that electrochemical gradient then can be used to make ATP. So there's two things going on here. All right, so these electron carriers in the electron transport chain, they're proteins. Most of them are proteins, or they are structures that are embedded in proteins, and they're all located in the inner mitochondrial membrane, right? So in total, this is what we've done with the first stages of cellular respiration, is that we've transferred food energy in our food, bond energy into reduced NAD, and then that is going to pass its electrons into the electron transport chain, and now we have oxidized NAD. So all that NAD is doing is it is taking the food energy from the electrons from our food and then passing it into the electron transport chain, and we use it again and again and again. Okay, we cycle. Eventually, all of these electrons down the chain are passed to oxygen. This is a half reaction. Let's change it, because I'd like that to be a whole we don't have half molecular oxygen, right? Okay, so change that to O2 plus 2 H2O. Okay, all right. And where did the hydrogens come from? Well, electrons, they came from the electron transport chain. And the hydrogens, we always have hydrogen ions out there, okay? And this will become clear here in a moment. All right, so this allows for a gradual release of that energy, because if we release that energy explosively all in one fell swoop, it would be too powerful to be able to make anything, right? It'd probably be destructive. All right, so this ATP synthesis, it is coupled to the exergonic transfer of electrons to oxygen. All right, so once again, the electrons go into electron transport chain, we pass them down, 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 and energy is used to do work to make electrochemical gradient. The electrochemical gradient then is coupled to the synthesis of ATP. All right, make it clear here. So this is a energy diagram sort of showing not all of these electron carriers, some of them. And this is put in the form of a graph where the axis over here is free energy, okay? The free energy level content of these electrons, all right? And so these electron carriers, they are in 4 protein complexes. And so they're joined together. Part of the upper part of the chain is in complex 1. There's complex 2 that's off on the side. And then there's some of the chain that is in complex 2 or 3, and some of the chain in complex 4. Now, between complex 1 and complex 2, 3, is a shuttle. Okay, here it's called Q, but it is called, the real name is ubiquinone. It is a soluble shuttle. Okay, it's in the membrane. Ubiquinone, okay? And so it shuttles electrons from the part of electron transport chain that terminates in complex 1 to the beginning of the chain, complex 3. And then between complex 3 and 4 is another shuttle, and that is cytochrome C, okay? Cytochrome C, all right? So this is how it's arranged. And notice here, now most of these carriers possess cofactors, they're proteins. Most of them are proteins, but they have non-protein additions that are necessary. And some of these, you don't have to memorize all this chain, okay? The only thing I want you to know is that there's 4 protein complexes that house this chain. I do want you to know that there is ubiquinone as a shuttle between complex 1 and complex 3, and I do want you to know that cytochrome C is part of the chain that serves as a shuttle transferring electrons between complex 3 and complex 4. I want you to know in general some of the cofactors that are in here. So some of these are flavoproteins, and I've already mentioned flavin. It's derived, it is a nucleotide derivative that is derived, nitrogen base that is derived from riboflavin. Some of these are iron sulfur proteins, so they contain as cofactors several iron and sulfur atoms. Some of these are cytochromes. Now what's interesting about these proteins, cytochromes, is they have a non-protein addition, which is a ring form, 4 rings with an iron ion at the middle, and this is called heme. I mentioned hemoglobin. Hemoglobin is a protein that has quaternary structures, 2 alpha units, 2 beta units, and its function is to carry oxygen. It also, each of those subunits has a heme, okay? So this compound is common to both hemoglobin carrying oxygen and cytochromes. All right, so these 4 protein complexes. Now, the mitochondrion originated from a gram-negative bacterium. So where in that gram-negative bacterium, the ancestor, would the electron transport chain be located? bacterium did not have mitochondria. It became the mitochondria. Okay, so where would be the electron transport chain there? Plasma membrane. Okay, plasma membrane. The innermost membrane of mitochondria is homologous to the plasma membrane of a gram-negative bacterium. The outer mitochondrial membrane is homologous to, or that means same origin, evolutionary origin, to the lipopolysaccharide outer membrane of gram-negative bacteria. Now, there's no peptidoglycan between these two, because that has disappeared, all right? It's been gotten rid of. However, that outer membrane has these beta barrel structures called porins. And the only place we find those structures is in the outer membrane of mitochondria, outer membrane of chloroplast, and the outer membrane of gram-negative bacteria, okay? Okay, all right. So now, this is an important enzyme. This very last complex four, let me back up here so you get some idea about how much protein is here. Each of these electron carriers with their cofactors are proteins, but they're not the enzymes. Between each of these steps, we still have an enzyme that catalyzes the passage of those electrons from one carrier to the next. The enzyme that takes the electrons from cytochrome A2, A3, and passes them on to oxygen is called cytochrome oxidase. This is an important enzyme because that is the enzyme that gets poisoned with carbon monoxide and hydrogen cyanide. And this is where that becomes a problem. So this is what the cytochrome oxidase does. In fact, once again, let's change this. Change this so we have 4 hydrogens, 4 electrons. So we have 1 O2 to produce 2 H2Os. Okay, this is a half reaction. That's what you see in the book, but that's not the reality because you usually don't have half an oxygen molecule. Okay, we have to do it all at once. Okay, all right. So there's 2 terms here: oxidative phosphorylation. This is a second way in which ATP is being produced. And so we're using the electron transport chain to create a electrochemical gradient and then the electrochemical gradient to produce ATP. And so this here is not substrate level phosphorylation because the origin of the phosphate that goes on to ADP is inorganic in the cell, okay, or in there. It isn't attached to a food molecule, and this is called oxidative phosphorylation. Why is it called oxidative? Because the food substrate, the energy comes from the oxidation of the food substrate, okay? All right, we'll get to it. Now, this mechanism was already proposed by Peter Mitchell back in the 1960s. That was a long time ago. And they didn't know much about what's going on here back then. What he proposed is that the inner mitochondrial membrane is impermeable to hydrogen ions. We've now, after our study and knowledge of biological membranes, that should be pretty clear. Okay, it wasn't so clear back then, all right? But anytime we have a charged ion, that has a hard time, almost impossible time, to get through a phospholipid bilayer, okay? So that's furthermore, this produces an electrochemical gradient, hydrogen ion gradient, okay? And that is generated by the passage of electrons through the electron transport chain, which mechanism I'll get to very much. In fact, looks like I have a link. We'll go ahead and do it, okay? So this is inner membrane space. This is the matrix, and this is complex 1, ubiquinone, complex 2, complex 3, cytochrome C, and complex 4. This is where the electron transport chain is. Now, I showed it as a graph before. Here it's in a more realistic scenario. But so as electrons, let's go back here. like I don't have, as electrons, NADH is in the matrix. And in fact, it's in the matrix, that's where the enzymes of the Krebs cycle happen to be. Okay, so things there are happening in the matrix. And so the NAD passes electrons into this electron transport chain, carried from complex 1 to 3 by euquinone, which is nonpolar. Okay, it's in the membrane. Electron transport chain through here, and we have cytochrome C is hydrophilic. It is in the inner membrane space. Okay, so it's not in the membrane, it's soluble, but it is usually close to the membrane, and it shuttles the electrons over here. Each time these electrons pass through complex 1, complex 3, complex 4, then the energy is used to move hydrogen ions from the matrix into the inner membrane space. Okay, so the loss of energy of electrons as they go down the electron transport chain is used to pump hydrogen ions into the inner membrane space, creating a hydrogen ion gradient. Okay, I'll come back to that in a moment. So that's an energy source. It's an electrochemical gradient. All right, and so how we connect that gradient to the production of ATP is that we have a special enzyme called ATP synthase that is also embedded in the membrane. This is an interesting protein because it has very much the same structure as hydrogen, ATP-driven hydrogen pumps for hydrogen ions, all right? I mentioned that before, how does a bacterium create electrochemical gradient, but it's flagellum. It has something that looks very much like ATP synthase. That is an ATP-driven hydrogen pump. This here is just the opposite. It is an ATP-driven, or it is an ATP pump going backwards. So if the pump normally moves hydrogen ions across the membrane using ATP, what's going to happen if it runs backwards? If we now use that hydrogen ion gradient to go the opposite direction, it's going to produce ATP. Okay, and that's exactly what happens here. All right, so that hydrogen ion gradient is coupled to the synthesis of ATP. As electrons move from the inner membrane space through ATP synthase, it causes a torque, causes a part of that protein to spin, and there is an attachment site for ADP and inorganic phosphate, and this energy is then used to make. Part of this is called chemoosmosis. Now, there's two words here, and they look very similar, okay? And sometimes it's very confusing. So oxidative phosphorylation, what is the difference between it and chemiosmosis? Chemiosmosis is just using a hydrogen ion gradient to produce ATP through ATP synthase. Oxidative phosphorylation, however, involves the electron transport chain too. Okay, so that's the process of passing electrons from your food to NAD to the electron transport chain and down to oxygen. Okay, and it includes chemiosmosis. Chemiosmosis is just that last step, using the using the hydrogen ion gradient to generate ATP. All right. Okay. So I should have. Okay, this is what ATP's cartoon what it looks like. So we have a high hydrogen ion concentration up here, and through the electron microscope on the matrix side of that membrane, you do see these big bumps. And we do know that when hydrogen ions going down their electrochemical gradient through a tunnel in this protein, that this here does spin. Okay, it creates a torque, spins it, and there is an attachment site there for ATP or ADP and phosphate, and it will generate ATP. So how much is produced? Okay. It takes about 10 hydrogen ions. Well, yeah, we get 10 hydrogens transferred per pair of NADHs to produce ATP. And we get 6 from NADH, all right? So, yeah, this is where I want to go to. Okay, because the experimental evidence is that for ATP synthase to produce 1 ATP, that it takes about 4 hydrogen ions going through the synthase. So since we are getting 10 of these, because how many NADHs have we produced total? How many do we get in glycolysis? 2. How many in pyruvate oxidation? 2. How many in Krebs? 6. How many? 6. So altogether, how many? 10. This is where the 10 comes from. You have 10 NADHs that pass these electrons through the electron transport chain, and but it takes 4 on average going through ATP synthase to produce ATP. So if we do this, we have 10 NADHs divided. So each passage of this electrons through the electron transport chain, we're ending up with 10 hydrogen, or 10 hydrogens getting pumped across. And so since we have 10 of them that came from NADH, takes 4, then on average it's 2.5 ATP per NADH. All right? Now, obviously you don't make half ATP, right? Okay, you either make an ATP or you don't, all right? So, but this here is based on an average looking at millions of operating systems here. And so sometimes this ATP synthase has to use 6 to make 6 hydrogen ions to make 1 ATP, sometimes 3, okay? This is just the average, stochastically. And this is a number also that I want you to know, okay? For NADH to produce ATP, we have 2.5 ATP that are produced per every NADH that we feed its electrons into the electron transport chain. Now, FADH, however, only contributes 6. Okay, when its electrons go through the electron transport chain, there's only 6 hydrogen ions that are pumped from the matrix into the intermembrane space. So therefore, it still takes 4 hydrogens, ions going through ATP synthase to make ATP. We only have 6 of them, so it's 1.5. So the electrons, in other words, let me summarize it. The electrons from 1 NADH is enough to produce 2.5 ATP on average. The electrons from FADH2 only leads to the production of 1.5 on average. Okay? Why is that? Okay, I'll go back to this. So once again, as we feed these electrons from NAD into complex 1, those electrons, as they go through here, they result in the pumping across to that complex of 4 hydrogen ions. Okay, this is a pair of electrons from NAD. A pair of electrons from NAD, as it passes down the electron transport chain through complex 1, it results in the movement of 4 hydrogen ions from the matrix into the intermembrane space. Then, those electrons will pass to ubiquinone into complex 3, and as we go through, those electrons go through the electron transport chain in complex 3, the result is the pumping from the matrix into the intermembrane space of 3 hydrogen ions. Cytochrome C carries it over to complex 4, and as those electrons go down the electron transport chain, ending up on oxygen, it results in another 3 hydrogen ions. Okay, so the pair of electrons from 1 NADH will result in 10 hydrogen ions being pumped into the intermembrane space. Right, that's where we had on Now, when these go back through ATP synthase down concentration gradient, the result, average result is 2.5 ATP. Okay, now why only 1.5 for FAD? FADH is not at as high of an energy level content. In fact, let me just go back. Go back far enough. Where's that figure? Went too far. Oh, going the wrong direction. That's the problem. Yeah, I want to go back to energy diagram. Yeah, right here. Okay, so NAD plus is way up here. It has sufficient free energy to pass electrons into the electron transport chain. But where FAD is, it is too low below. It can't pass the electrons into the top of the chain. It doesn't have enough free energy. And therefore, the entry for its electrons is complex 2, okay, at a lower level. So those electrons don't go through complex 1. They go through complex 3, which leads to 3 hydrogens pumped across, and complex 4, 3 hydrogens pumped across, only 6. Only 6. Those 4 those 4 hydrogen ions that are pumped as electrons pass through complex 1 doesn't happen with FADH2. And therefore, we only get 6. Divide by 4, because it takes 4 hydrogens to produce ATP, we get 1.5. Okay? So these are numbers I want you to know, that how many ATP are produced per NADH. Okay, 2.5 for NADH, 1.5 for electrons of FADH2. All right, so put it all together. All right, so how many ATP can we produce altogether? So we had 10 NADHs that we produced in total when we start with glucose. And if each of these, the pair of electrons from NADH results in the production through chemiosmosis of only 2.5, then that gives us 25 ATP. FADH2, we only produce 2 of them in the Krebs cycle, that's all. And they produce their electrons going down the electron transport chain, pumping across hydrogen ions, and those hydrogen ions then passing through ATP synthase, only enough to produce 1.5. So we have 3. And then from glycolysis, by substrate level phosphorylation, 2 ATP. From Krebs, we also had 2 by substrate level phosphorylation. These up here, 28 of the ATPs are produced by oxidative phosphorylation. The vast majority of ATP is produced by oxidative phosphorylation. Only 4 ATP are produced by substrate level phosphorylation. So you should see how this was a good mutualistic relationship, because the ancient alpha proteobacterium from which mitochondria actually evolved, the earliest eukaryotic cell probably only had glycolysis. That's all. It could at most produce just 4 ATP per cycle. But having a symbiont in it that has electron transport chain, can use oxidative phosphorylation, it greatly increased its ability to produce energy with ATP. Yes?
So oxidative phosphorylation, is it, what is it exactly? So
It's the stripping of electrons from food to NADH, FADH2, and sending of those electrons down the electron transport chain.
so
And the production of ATP by chemiosmosis, okay, through ATP synthase. So oxidative phosphorylation includes more than just chemiosmosis. Chemiosmosis is having electron gradient, hydrogen ion gradient, and producing ATP through ATP synthase. So oxidative phosphorylation also includes the steps of stripping the electrons from the food and sending them down the electron transport chain.
So it would include something like after Krebs cycle on how, like, so it includes all the electron transfer
Yeah, all of it,
from like glycolysis.
from the
to
food on down.
Krebs. Okay.
Yeah. Okay, so we get 32 ATP. All right, now when I asked you to calculate if all this 686 kilocalories per mole of the complete oxidation of glucose could be captured, what did we come up with?
90?
Something like 90 something, okay? This is the reality. Okay, this is what we actually get. So what happened to the rest of the energy? He's lost his heat, okay? So you calculate the efficiency. You calculate the efficiency, okay? I'm not going to tell you how to do that. So what am I asking? How much of that 686 kilocalories per mole actually ended up in ATP? That will give you the efficiency, all right? Because I might ask about that. Okay, all right? All right, so now, this is the trick in animal heart, liver, kidney. If there's no other competing processes going on, we get 32 ATP under standard conditions from 1 glucose. However, in other cells, other than heart, liver, and kidney, it's reduced to 30. Why is that? Okay, where is the top of the electron transport chain located? Exactly, what compartment is it? Where is the top of the electron transport chain? It's inside the myocardium, right? On the matrix side of the inner membrane. 2 of our NADs were produced in glycolysis. Where's that? Cytosol. Okay, those electrons have to somehow get across the outer membrane, the inner membrane, to the matrix. Okay, that getting from the cytosol into the inner membrane space works, but not across the inner membrane. Okay, so there is shuttle systems, a protein system. I don't have time to talk about it. I'll leave you in suspense. There's a shuttle system in the inner mitochondrial membrane whereby the electrons from NAD are passed to an NAD on the inside. But there's 2 different types of shuttles. Type of shuttle in heart, liver, kidney cells that can pass those electrons without loss of energy. Those in the shuttle system that's in the mitochondria and other organs, it costs. And so there's a cost equal to 1 ATP value to get those electrons into the electron transport chain. And since we produce 2 NADHs in cytosol, it represents a loss of 2 ATPs. And I'll explain that on Thursday. Stop here. It's a good place to stop. Okay, so signing papers, bring them up. Make sure everyone's